Happy Monday. So, first announcement, exam on Friday. Exam is in this room at the regular time. I had several questions about that. Yes, exam is on Friday in here at noon. There are some logistical things that will be necessary in order to make this work. First of all, I'm not going to be here. So, somebody else will be giving the exam, and it's going to take your cooperation to make that happen. So I'm going to say some of the instructions here today. I'm going to repeat them on Wednesday. So if you follow instructions, you'll have more time to do the exam. And the more time that the TAs and the faculty member have to spend rearranging you, the less time you'll have for the exam. Everybody will start the exam at the same time. Okay? So here's how we're going to do it. First 10 rows, starting at the front, you can sit in every seat. Okay? First 10 rows, you can sit in every seat. After the first 10 rows, you will sit in every other seat, and here's how you will do it. You will start numbering this seat as number 1. Then you'll skip a seat, and then you'll go to the next seat as number 3, right? So you're going to sit in 1, 3, 5 as appropriate. And if you decide you can't do a left-handed or a right-handed or whatever, then go somewhere else. Don't shift because you've got the wrong seat, wrong-handedness that you want. I'm left-handed and I've used right-handed seats forever, so it's not a big deal. But make sure that you sit like that, 1, 3, 5. These guys will sit 1, 3, 5, 7, 9, 11, etc. If somebody is sitting in 6, don't scoot over to 8. Okay? Sit in 7. You might remind them, hey, you're sitting in an even-numbered seat. You're going to need to move because they're going to make you move. Okay? And then last, this one over here. And this is true on the top as well as the bottom. Okay? 1, 3, 5. Okay? So first 10 rows down here in front, every seat's fine. Okay? Everything up above and everything past the first 10 rows, every other seat. Are we clear? Okay, like I say, the more time we have to spend shuffling people, the less time you'll have to do the exam. They will work to get the exam out quickly, okay? But your cooperation is obviously very helpful. Second, I will do a review session for the exam, and that will be tomorrow evening at 5 p.m. in Gilfillan Auditorium. I will videotape it. Tomorrow, 5 p.m. in Gilfillan Auditorium. I haven't decided yet where the exam material will end. Okay? It probably will go into Wednesday. It probably will go into Wednesday, but I'll announce on Wednesday where it's going to stop. Okay? All right, those are the two biggie things. All right. Well, last time when I finished talking about the material, I had just gotten to this plot, which is a plot that we call V versus S, which is velocity versus substrate concentration. V versus S plots arise in the way that I described to you last time. I take about 20 different tubes, all of which have buffer, all of which have the same amount of enzyme, and each of which has a varying amount of substrate. Okay? Now somebody said, why don't I vary enzyme and substrate? And the answer to that is, I can't do an experiment where I've got two variables here. I've got to do an experiment with one variable. The one variable that I have is substrate. If I do that, then I see a plot that looks like this. I see that initially the plot rises fairly rapidly. This is a hyperbolic plot like we saw with the myoglobin binding of oxygen. It rises fairly rapidly, and then it starts to fall off. I shouldn't say fall off, but level off. It doesn't fall off, but it levels off. All right? That leveling off is happening because the enzyme is getting saturated with substrate. All right? So the analogy is the factory. If I add more raw materials when a factory is already at capacity, it's not going to produce any more. The raw materials are just going to accumulate. Same thing is happening here. As I get up close to the point of saturation of the enzyme, it's not going to go beyond a certain point of velocity. That point of velocity is what we refer to as maximum velocity. You see on this graph that we have to sort of guess where that leveling off place is. I'm going to show you later a different kind of plot that allows us to determine the maximum velocity pretty precisely. Okay. All right. Let's see, where was I? Okay. Those are not a surprise. At the active site, the substrate has to bind. And the binding of the substrate at the active site is not a covalent binding. At least it's not a permanent covalent binding. We will see when we talk about enzyme mechanisms after the exam that there are sometimes covalent intermediates that are made, but their bonds are broken. Okay? If we formed a covalent bond with the enzyme that didn't get broken, we would have killed the enzyme. And this is actually a strategy of some drugs. Penicillin, for example, works that way. It binds to an enzyme that bacteria need to make membranes, and when it binds to it covalently, the enzyme is dead. Okay. Now, I want to say, before I talk about some of the kinetic parameters, I want to say a little bit about enzyme mechanisms. Okay? Enzyme mechanisms. We talk about two different mechanisms that are commonly discussed with how enzymes work. And it's important that you understand the difference between these. The most common one that you hear is the one that you learn about in high school and you learn in basic biology, and that's called the Fisher lock and key model. The Fisher lock and key model. And most people think, most people who don't know think that's how an enzyme works. They've given the analogy of the enzyme is like a lock and the substrate is like a key, and the key fits into the lock precisely, and the enzyme gets that binding is really what's happening. If you have the wrong key for the lock won't take it and nothing will happen. Okay? Well, the Fisher lock and key model is very good at explaining the specificity of binding of substrate. By specificity, I mean the specific binding of a substrate. We know that substrates usually have a very specific structure. So it explains the specificity very nicely by the analogy between the key and the lock, but it doesn't tell us anything about how the enzyme is doing what it does. So the Fisher lock and key mechanism is not a good mechanism for explaining how catalysis works. You see the Fisher lock and key on the screen here. Here's the enzyme, here is a specific binding site, and oh my goodness, it exactly matches the substrate, so they bind, and it forms this thing called the ES complex. This ES complex is where the reaction occurs. Okay? A better model is what's called the Koshland-induced fit model. Okay? The Koshland-induced fit model is much more accurate for explaining how an enzyme actually does what it does. You see in this model that the enzyme has a binding site that looks sort of like the substrate, but not exactly like it. And you see that after the substrate has bound to the enzyme, that the enzyme has actually changed shape. It has gone from looking like the structure on the left to looking like the structure on the right. Now, the binding of the substrate changes the enzyme. That shouldn't come as a surprise to you because you've seen other proteins like myoglobin and hemoglobin whose structure changes as a result of binding of substrate, or binding, in that case, of a molecule. Substrate is only with an enzyme, but binding of a molecule. Okay? Enzymes change their shape on binding to a substrate. Now, that change of shape actually turns out to be of great benefit, and it also helps to explain why enzymes work so efficiently, so fast, so rapidly. Okay? The enzyme we can think of sitting over here is just kind of lolling around, right? It's in a very relaxed state. When it binds to the substrate, we could imagine that the enzyme is now in some way strained. Strained, okay? The strain of that enzyme is communicated to the molecule. Let's imagine that this enzyme catalyzes a reaction where ABC breaks apart into A and BC, right? In other words, we're breaking a bond right here between A and B. If the enzyme is under strain, it's going to be much more likely that that strain is going to relieve by the breaking of the bond. That's just one example. There are many examples that we can have where the structure of an enzyme is changed by the binding of substrate, and that change helps the enzyme to do what it does. We'll talk later in the term about a very interesting enzyme called hexokinase. Hexokinase catalyzes the first reaction, the breakdown of glucose. And it has a structure that we don't have to worry about too much, but I'm going to show it to you schematically that looks like this. Hexokinase, what it does is it catalyzes the transfer of a phosphate from ATP onto glucose. It transfers a phosphate from ATP onto glucose. That means that the enzyme has to bind to both ATP and to glucose. Everybody with me? When this enzyme binds glucose, okay, so here are the two binding sites. Here's a binding site for ATP. Here's a binding site for glucose, okay? When this guy binds glucose, all right, the enzyme now becomes very favorable to bind ATP. The structure has changed slightly, and now ATP binding is favored. When ATP binds, the enzyme goes through a structural change that causes the glucose down here to be brought into close proximity to the ATP here. We can think about it as two jaws that go like this. Now, the phosphate of the ATP is right next to the glucose that is being transferred to right here. In this case, it's very easy to see how that flexibility of the enzyme and the change of shape on the binding of substrate facilitates the enzyme catalyzing the reaction. As I said, there are many examples, but these are probably two of the most vivid that you can have for the changing shape of an enzyme. In summary, what I say about the Koechlin-induced fit, which is what I'm talking about in all of these mechanisms, what I can say about the Koechlin-induced fit is that the enzyme changes the substrate. But the substrate transiently changes the enzyme. Now notice I said transiently, because I've already talked about how these jaws can come together and how the reaction can be catalyzed to where the phosphate is transferred from ATP onto glucose. When that reaction occurs, you have down here glucose 6-phosphate, and on top you have ADP. These are different from what the enzyme started with. They're not ATP and glucose. They're glucose 6-phosphate and ADP. And you could imagine that they're going to cause the enzyme to have a shape change as well, and they do. The jaws open. The jaws open, ADP goes out, glucose 6-phosphate goes out, and the enzyme is left back in its original state. It's now ready to bind a new ATP and a new glucose and repeat the process over and over. So, I'll repeat that. An enzyme changes a substrate, okay? And a substrate transiently changes an enzyme. If we were to describe the Fisher model, we would say that an enzyme changes the substrate, but we don't know how. Because the Fisher model doesn't tell us anything about how the enzyme works. Questions about that? Okay. Good. All right. Well, let's talk about velocity. Okay? Let's talk about velocity. Let's imagine I've got a reaction that I start. I've got an enzyme I've got my substrate. I start the reaction. And when I start the reaction, I only have substrate. I have no product. Remember, the enzyme is going to convert substrate into product, right? When I start the reaction, there's no product. Which direction is the reaction going to go? Forwards, right? Because there's no product, the only way for it to go is forwards. As product starts to accumulate, what happens is the delta G value will start to become more positive after starting out negative, right? And it means that we're going to start seeing more reverse reactions. If we let the reaction go long enough, the enzyme will be at equilibrium. And when a reaction is at equilibrium, I hope you remember this, it means that the forward reaction equals the reverse reaction. We don't see a change in concentrations of substrate and product once we reach equilibrium. Okay? So equilibrium means forward rate equals reverse rate, concentrations do not change. Well, when I'm interested in studying an enzyme reaction, I'm not interested in studying equilibrium because that's zero velocity. I want to know how well does this enzyme work. It'd be like taking a racing car and deciding to measure its velocity when it's parked in the parking lot. It wouldn't tell you anything, right? To measure the velocity of an enzymatic reaction, I need to measure what we call initial velocity. That initial velocity occurs before the substrate concentration starts to accumulate. The more it accumulates, the lower my estimate of the velocity is going to be. So when we talk about the measurement of velocities, like we saw on the V versus S plot, those were all initial velocities, meaning that we measure the velocity very quickly before the reaction gets too far. Okay? We could measure it a few seconds. We could measure it a minute. It varies. But the point is, we don't want to wait an hour and the reaction is at equilibrium because we're not going to get any velocity if we do that. Everybody with me? Okay. So initial velocities are very important. You can see equilibrium occurring here. And I won't emphasize that figure too much because I think that figure is a little confusing. All right. So the velocities that we measure are initial velocities. By the way, what I'm talking about here, all of the kinetic parameters that I'm talking about, velocity, and the other ones that I'll be talking about today, are part of what we call Michaelis-Menten kinetics. Michaelis-Menten kinetics. If this were the majors class in biochemistry, I would be going through the derivation of Michaelis-Menten kinetics. I'm not doing that here. I don't think it's necessary for you to memorize a derivation. I think it's much more important for you to understand the parameters that we use in describing and understanding enzymes. You've heard the first one, and the first one, of course, is maximum velocity. The next one I want to talk about is one that's a little harder to get your heads around, but it's not overly complicated. It's called Km, or the Michaelis constant. Okay, you see it right here. The Michaelis constant, I'm going to tell you what it is and how we get it, and then with some considerations about it. What it is, in simple terms, is a measure of the enzyme's affinity for its substrate. Affinity. What is affinity? Affinity is the desire to bind to and hold on to. It's like what you have with your significant other, right? Unless you've had an argument, in which case you probably have low affinity for your significant other. But if things are really rosy and it's Valentine's Day, you are having a nice affinity moment with your significant other, and that's what enzyme has with its substrate if its affinity is high. Enzymes vary in the affinity that they have for their substrate. They vary. Okay? But a given enzyme will have a given affinity for a given substrate. That is, it's like a constant. It is a constant. A given enzyme will have a fixed affinity for a given substrate. Now, how do we get that measure of affinity? Well, you could say, well, if I want to measure the affinity of the enzyme for the substrate, I might measure the velocity because the affinity in some way could be related to the velocity. And the reality is it's not, although it looks like it is. It's a little confusing. There's not a direct relationship between affinity and velocity, but there is with velocity over 2. Whoa, what does that mean? Okay. If you think about it, maximum velocity is one type of velocity. And maximum velocity occurs when? When? When the enzyme is saturated with substrate. In theory, maximum velocity occurs at infinite substrate. In theory. Well, if we take everything to maximum substrate, we don't really get a very good way to measure how much the enzyme is binding substrate, right? Because there's an awful lot of enzyme that's not bound to substrate. Instead, we measure the affinity by measuring the substrate concentration that it takes to give the maximum velocity over 2. Maximum velocity over 2 does not take an infinite amount. And as you can see on this plot, it looks something like this. There is a way of measuring affinity by measuring Vmax over 2. Now, there's still no relationship between velocity and Km. That is, Vmax over 2 could be high, it could be low. Km could be high, it could be low, and they're not connected to each other. But this is the way that we measure them, is by doing this. Let's imagine, thinking about Km, that I told you that I have an enzyme that has a low affinity for its substrate. A low affinity for its substrate. That would mean that they've had the argument, right? The enzyme doesn't want to bind to the substrate and is only going to bind to the substrate when the substrate comes knocking on its door. Is this enzyme going to have a high Km or a low Km for its substrate? It will be high. It will take a lot of substrate to get the enzyme to wake up and get to half maximum velocity. On the other hand, if an enzyme has high affinity for its substrate, it's going to be seeking it out. It's going to be grabbing it wherever it can, maybe in embarrassing moments walking across campus. Okay? But it's going to be grabbing it wherever it can. Okay? It's going to have a low Km. So Km is inversely related to affinity. High affinity, low Km. Low affinity, high Km. Now I'm going to tell you something that a third of you are going to put on the exam and you're going to get wrong. And even though I'm going to tell you, you're still going to get it wrong. That always bothers me, but it's true. I'm going to ask a question on the exam that's going to be related to Vmax or Km or something like that. Vmax over 2 or Km, okay? And a third of you are going to tell me that Km is Vmax over 2. I want to point out Vmax over 2 is this point, and Km is this point. Km is a substrate concentration. Vmax over 2 is a velocity. It's like saying the price of gasoline versus the speed that you drive the car. They're not the same thing. Okay? Km is not Vmax over 2. Km is the substrate concentration that gives Vmax over 2. Very different things. A third of the class will get that wrong. I don't know why, but you will. Don't get it wrong. Make me happy. Okay. Well, what you've seen was, first of all, we had to estimate where that Vmax plot was, where that line flattened out, and we're not exactly sure where. And because we're not exactly sure where Vmax is, we're not exactly sure where this Vmax over 2 is, so we have a hard time finding Km. We would like to find an easier way to do that. an easier way to do that, and we have one. It's a different kind of a plot called a Lineweaver-Burk plot. Lineweaver-Burk. Okay? Lineweaver-Burk plots use the same information as the V versus S plot. The 20 tubes, the 20 substrate concentrations, and the 20 different velocities that we get. So why don't they look the same? Because we invert all of the data. All of the velocity measurements we take as 1 over the velocity, and all of the substrate concentrations we take as 1 over the substrate concentration. And then we plot 1 over the velocity versus 1 over the substrate concentration. And when we do that, that hyperbolic plot that we saw on V versus S becomes a very nice and simple straight line. That straight line has 2 points on it that are very easy to understand. One of those is the place where the line crosses the y-axis. That has the exact value of 1 over Vmax. And the place where the line crosses the x-axis is equal to minus 1 over Km. Thanks to this plot, I can determine those numbers quite readily, and therefore determine the Km and the Vmax very easily. So Lineweaver-Burk plots are very, very helpful in allowing me to measure these parameters that are of interest to me. Questions about Lineweaver-Burk plots? There usually are. Yeah? How did I know that just by inverting those? Well, mathematically I could know that, but you don't even have to know that. You simply know that if you do that inversion, it will turn out linear. You can do the numbers yourself and see. I think there's one of the problems in the book where you actually do that, and you'll see how that works. It's kind of cool. Oh, yeah. Yeah. Is there a way to tell when they're at equilibrium? You don't want them at equilibrium. Because remember, if you let them go to equilibrium, you have no velocity. No. You're measuring velocity versus substrate concentration. The velocities you're measuring are initial velocities. You are definitely not measuring conditions at equilibrium. Okay? Very important point. It's a good question, but these are not at equilibrium. No. Other questions? How about a limerick? Excuse me for feeling superior to all the low bacteria. Students know very well there are no organelles inside their tiny interior. Okay. I like Ogden Nash. That's kind of an Ogden Nash poem. Anybody know Ogden Nash? Or am I just that old? I am that old. You guys know, but you weren't going to say that, right? It's just reminding me I'm that old is what it is. All right. Here are some enzymes, and here are some Km values. And no, you don't need to know these. I'm just showing you as an illustration of some enzymes and the Km values they have for various substrates. Okay? Some of these range from very high, and by the way, these are in units of concentration because remember that Km is the concentration that gives Vmax over 2. These are all concentrations. We could look at something like chymotrypsin with its value of 5,000 micromolar and compare it to arginine tRNA synthetase, which has 3 different Kms. Whoa, what's going on with that? What do you suppose happens with that? How can it have 3 different Km values? 3 different substrates. So remember I said that it has a fixed Km for a given substrate. These guys have different shapes. They bind at different sites. And so the enzyme, not surprisingly, has different affinities for them. But in each case, the affinities of the enzyme for any of these is greater than the affinity that chymotrypsin has for this substrate shown on the top. Now, there's one more parameter I want to tell you about that may make more sense to you. Okay? So let me tell you the parameter, and then I'll show you a very cool example of that. Okay? The parameter I want to talk about is something called the turnover number. Now, you might have looked at the Vmax and decided that, wow, Vmax tells me a hell of a lot about an enzyme because I know how fast the enzyme goes. And I'm going to tell you that it really doesn't. After all this time I spent telling you about Vmax, now I'm going to tell you it doesn't tell me enough about an enzyme. Let's go back to the factory analogy that I made for you before. I've got a factory that's producing automobiles, and it's got all the raw materials it needs. It's saturated. It's producing 300 cars a day. And it doesn't matter how much more raw materials I've got, the maximum rate that the assembly line can put them out is 300 cars per day. Well, if I'm the president of General Motors and I say this is a really popular car, and we need to get as many of these cars on the lot as possible, the only way I can make more cars per day is if I make another factory. So I make an identical factory to the one that was producing the 300 cars per day, and it starts producing 300 cars per day, and now I've got 600 cars per day. Everybody with me? So 300 cars per day only told me something about the one factory. My question to you is, how many of you know how many factories GM uses to produce a given car? You probably don't know, right? You probably don't know how many factories GM is using to produce a car. I give you this analogy because when you do an enzymatic analysis and someone says, I had a Vmax of 300 micromolar per second, okay? You say, that's great. How many enzymes did you use? Just like you would say, how many factories produced it? Without the information about how many enzymes were involved, the Vmax doesn't tell you anything. If I double the amount of enzyme, I'm going to double the Vmax. So Vmax is useful only when I know the amount of enzyme. So this is where we get into turnover number. When I know the amount of enzyme that I use, I can take the Vmax and divide it by the concentration of the enzyme. So remember that Vmax is concentration of product per time. So I've got a concentration of product that I'm dividing by a concentration of enzyme. Concentration cancels concentration. And I get a number. I get a number per time. I'm going to repeat that. I take the Vmax, which is concentration of product per time, divided by the concentration of enzyme, and I get what we call the turnover number. It is a number, and it's a number per time. An example might be that I could have an enzyme that has a turnover number of 1,000 per second. 1,000 per second. That would be a turnover number, and what it would mean is that every molecule of enzyme in the reaction is producing 1,000 molecules of product per second. Every single enzyme is producing 1,000 molecules of product per second. Turnover number turns out to be a very, very useful number that I can compare between enzymes because I don't have to worry about the amount of enzyme because I've already taken it into consideration. Turnover number has what we call a turnover number we name Kcat. The letter K with the subscript CAT for catalysis. Okay? Let's look at some turnover numbers. When I say enzymes are as close to magic as we get in biochemistry, this is one of the examples I frequently show. Look at carbonic anhydrase. It has a turnover number of 600,000 per second. That means, to repeat myself, That means that every single molecule of enzyme is producing 600,000 molecules of product every second. There's nothing in the macroscopic world that we live in that is anything like that. Nothing. Okay? It's incredible. I mean, even 100 per second is remarkable. But we're talking about 600,000 molecules of product per molecule of enzyme per second. Amazing. Okay? We're going to see in a minute that for some enzymes, they are so efficient at what they do that we describe them as perfect. They have evolved to the point where they cannot be improved further. What does that mean? They cannot be improved further in terms of their efficiency. Any mutation that they have will make them less efficient. And the reason that we know that they're perfect and they're at their maximum efficiency is that we know in an aqueous solution how fast diffusion can occur. The substrate has to diffuse into the active site. Perfect enzymes are limited only by the rate with which the diffusion of the substrate can make it into the active site. That's the only limitation that they have. That's why any mutation that they suffer causes them to be less efficient, and they will never get more efficient than that limitation of diffusion. That's remarkable. How do we get a perfect enzyme, or how would we describe a perfect enzyme? Okay? If I go back, if I go back to this guy up here, this was the plot I showed you to begin with. If I look at this plot and I say, wow, there is the turnover number, okay, which is in kcat, all right? Actually, it says kcat per second, but it's it is per second. That's right. So kcat is in units of per second. We can see various enzymes that are shown here. Carbonic anhydrase here is shown at about 1 million. And by the way, kcat will vary slightly with the conditions that you use it under. Okay, but we think of it as essentially a constant. It's at about 1 million. Here is the Km value. Where's the Km? Oh, blast it. I thought I had it on there. Sorry. Here it is. Okay, so we take something like carbonic anhydrase, and we take the kcat, which is that 600,000 or that 1 million number that I was talking about earlier, and we divide it by the Km. Now let's think about this. A totally efficient enzyme would have a kcat that's high. We all agree with that, right? How about the value of Km? High or low? Low. It should take very little to get it to half Vmax. So the larger this ratio is of kcat to Km, the more efficient an enzyme is. These guys all right here in the order of 10 to the 8th, 10 to the 9th are at their maximum efficiency. They can't go any faster. They can't bind the substrate with any better affinity, and they can't make the reaction rate go any faster. But they are working at maximum efficiency. These are examples of enzymes that we call perfect enzymes. Now, one of the questions I frequently get from students at this point then is, well, if evolution is so great, okay, how come all enzymes aren't perfect? Yeah, right? The answer is you don't want them to be. Just like you don't want everybody having a Maserati to drive 100 miles an hour to Fred Meyer's, you don't want all your enzymes to be perfect. And the reason for that is Maseratis going 100 miles an hour to Fred Meyer get into accidents, run over people, cause problems. Enzymes that are going too fast and out of control have the same problem. Only a handful of enzymes are perfect. And they're perfect because, well, actually there's a couple of reasons why they are, but one of the reasons in which they're perfect is because they are working so fast to prevent an unstable intermediate from forming, which can happen also fairly rapidly. Okay? In other cases, speed is of the essence. And getting that speed going really makes sense. Most enzymes are not perfect because cells, as we shall see soon, need to exert control over enzymes. If cells do not exert control over enzymes, cells are dead in the water. Okay, questions about that? A lot of material here today. Yeah? What places in the body would you have that? Well, these are in every cell of the body. Okay, so question is, give me maybe an example. I'll give you an example right here. This is one we'll talk about later this term, triose phosphate isomerase. Okay, triose phosphate isomerase is an enzyme that catalyzes a very important reaction in the breakdown of glucose. Okay, in the breakdown of glucose. The mechanism that this enzyme uses results in an intermediate that, as I mentioned, can be very unstable, and if it's released, will either cause a problem in the cell as a poison, or it will covalently bind to the enzyme and kill it. So the enzyme is perfect because it's binding the substrate as quickly as it can, as efficiently as it can, and its reaction rate is so fast that it doesn't get a chance to accumulate the unstable intermediate. So that's one example. There are other examples that cells can use to overcome problems as a result of their substrates, for example. That help? Yes?
You mentioned earlier that the perfect enzymes are limited only by diffusion of substrate.
Yes.
And I was confused as why it's aqueous.
Okay, so the question is, I said earlier that the perfect enzymes are limited only by the diffusion of substrate to the active site, and his question is, why do we only worry about it in an aqueous solution? Because if we have something besides an aqueous solution, the enzyme will denature. So it's got to be in an aqueous solution in order for this measurement to occur, because our cells are aqueous solutions. Okay? Yes? What does the diffusion of the substrate to the active site really mean? It means literally just that, how fast the substrate can get into the active site. Just how fast it can get in there. Now, when you think about this is, like I said, this is the magic part, and the magic part really isn't the enzyme. The magic part is the diffusion. 600,000 of these diffuse into the active site per second. It tells us that the nanoscopic world is very different from the world that we see out here. We cannot imagine 600,000 things happening in here in 1 second, right? Well, I can't. Maybe you guys can, but I can't imagine that, all right? For something like that to happen, and the logistics through which that occurs, it says that diffusion is something that we don't have a real good, real-world handle on out here. But that's what's happening with the diffusion of that. Yes? Does it have multiple active sites? We'll see that some enzymes actually do have multiple active sites. Yes, not all, but some do. Yeah. A situation where you would want an enzyme not to be perfect? Most enzymes are not perfect because they're more easily controlled. So if I have a good example we'll talk about later this term is the breakdown of glycogen. Glycogen is a polymer of glucose. And we will see that the breakdown of glycogen, even with a relatively inefficient enzyme, can occur very rapidly. We can deplete our glycogen stores quite readily, and glycogen is very important because what glycogen gives us is backup energy for muscles. Our liver's full of glycogen, our muscles are full of glycogen, and you'll see at the end of the term that we can turn on the breakdown of glycogen very quickly, release a lot of glucose, and we also have to be able to turn it off very quickly as well. If the enzymes that break down glycogen were more efficient, even if we raced to get them turned off, it would not work well. One of the mechanisms for turning them off is hormone action. Hormones are released in one part of our body, travel in the bloodstream to another part of our body, and can take seconds to minutes. If we have a perfect enzyme breaking down glycogen, by the time the hormone got to its target, we would have no glycogen. Does that make sense? Okay, good. Up top, yeah, okay. You don't understand how the products aren't going to be a limiting factor. Okay. Okay, so it's a very good question. His question is, won't accumulation of product be a problem? And the answer is yes, it can be. It can be. So having something that reaches a very rapid equilibrium, which is really what this is doing, okay, is good and effective. That's one thing, one consideration. And the other consideration is when we talk about reactions, they're not dead ends usually in the cell. They form the substrates for another enzyme for the next product, and the next product, and the next product. So it's not a simple picture as we will see, but it's a very good question. Well, we've talked about enzymes. Let us sing a song about enzymes. This is the most popular song that I've ever written. It's called Enzymes. I hope you'll sing it loud because it's a fun song to sing. When I get to the part where I raise my hand, I want everybody to jump up and say enzymes really loud, okay? Reactions alone could starve your cells to the bone. Thank God we all produce enzymes. Units arranged to make the chemicals change because you always use enzymes. Sometimes mechanisms run like they are at the races. Witness the cat of the carbonic anhydrases. How do they work? Inside of the active site, it grabs onto a substrate and squeezes it tight in an enzyme. Catalysis in an enzyme. V versus S in an enzyme. All of this working for you. Enzyme, enzyme. Energy peaks are what an enzyme defeats in its catalysis. Enzymes. Transition state is what an enzyme does great, and you should all know this. Enzymes. Catalytic action won't run wild, don't get hysteric. Cells can throttle pathways with an enzyme allosteric. You know it's true. So when an effector fits, it will just rearrange all the subunits inside an enzyme. Flipping from R to T, enzyme. Slow catalytically, enzyme. No change in delta G, enzyme, enzyme. You should relax when seeking out the Vmax, though there are many steps. Enzymes. Lineweaver-Burk can save a scientist's work with just 2 intercepts. Enzymes. Plotting all the data from kinetic exploration lets you match a line into a best-fitting equation. Here's what you do. Both axes are inverted, then you can determine Vmax and establish Km for your enzymes. Sterically holding tight enzymes. Substrates positioned right. Enzymes inside the active site. Enzymes, enzymes. All right, guys.